Okay, let's pick up from where we left it, and that was in an energy production pathway known as glycolysis, this anaerobic methodology of producing ATP, anaerobic meaning without oxygen. We found that it does indeed produce a small amount of ATP per glucose molecule. There were problems with glycolysis, which made it an unsustainable sort of thing. If it was a sustainable sort of energy production pathway, we wouldn't need oxygen. We could live underwater and we wouldn't have to breathe. But we do, and that is the result of another energy production pathway, which we'll talk about shortly. The problems with glycolysis, as glucose is being catabolized by glycolysis, is number one, we yield a lot of pyruvate, that 3-carbon pyruvate molecule, which can build up to toxic concentrations within the cell unless it is gotten rid of, meaning converted to something else. We also cycle a lot of NAD to NADH. So in the production of this pyruvate and the reduction of NAD to NADH, we have an unsustainable sort of situation. We have to have a good store of NAD to convert to NADH in order for glycolysis to continue, and we also have to have a good amount of pyruvate, or get rid of pyruvate rather, into something else. Now the way that we did this, at least temporarily, bring down the pyruvate concentrations and cycle NADH back to NAD was through fermentations. And there was two pathways. The one that's looked at most of the time, and is the major one, is the lactate fermentation pathway. Again, we're getting rid of pyruvate and converting it to something else, lactate. And we're cycling NADH back to NAD, which is both of the things we want to do. Now we're bringing down concentrations of pyruvate and we're cycling NADH back to NAD. The other minor fermentation pathway is alcohol fermentation. Again, what that ultimately does is it cycles pyruvate to another molecule, in this case ethanol, and we cycle NADH back to NAD so that glycolysis can continue. Both of these things are necessary in order for glycolysis to continue. But let's think about this now. What we're doing is, as I said before, it is unsustainable. If we look specifically at lactate fermentation up on the top here, yes, we are getting rid of pyruvate. We're converting it to lactate. Now, so here's our pyruvate concentration slowly reducing. That's a good thing. But in so doing, that means our lactate concentration is increasing. So we're basically running into another problem of high metabolite concentrations within our cells, which can ultimately be problematic. Thus, glycolysis, while it can be extended due to fermentation, is unsustainable. And it is at that point where, thankfully, we have another type of energy production pathway, which is aerobic, which can actually take care of all of these problems within that we see here with our glycolysis. Now, so glycolysis is used exclusively. Let's think about a situation, we talked about this last time, where glycolysis is being used exclusively. And that would be under conditions of strenuous exercise, like sprinting as fast as you can for as long as you can. And we all know we can't do that. We eventually fall down in a quivering heap and we are done. That means we have reached the end of our possibility for glycolysis, and that our more efficient energy production pathway that requires oxygen, which we'll be looking at shortly, simply can't get the oxygen to the cells quickly enough, so we have to depend completely on glycolysis, which, as we know, is unsustainable. So let's pick this up where we have just completed strenuous exercise. We sprinted as far as we can, as long as we can, and we're done. Glycolysis has reached its limit. Fermentation has reached its limit, and now what we ultimately have are a lot of 3-carbon metabolites which need to be cleaned up from our system. To do this, we have to allow our second energy production pathway to take over. More on that later. But let's take a look at what ultimately happens to all of these 3-carbon metabolites of glucose. Basically, I'm talking about pyruvate and lactate. We're going to start with a series of 3, I call them G words. 3 G words. They start with G, and they kind of sound alike, but they mean very different things. So let's take a look at our first G word. Our first G word is called gluconeogenesis. Gluconeogenesis. It is the synthesis of glucose in animals. Now, normally, this is really a very foreign and strange thing for animals. Animals typically use glucose. They catabolize glucose to extract the energy contained within the chemical bonds from it. It is plants which produce glucose typically. So we're doing something in gluconeogenesis that doesn't happen all the time. It only happens when it must happen. And when it must happen, what we're looking at are these 3-carbon metabolites here, pyruvate and lactate, are building up to very high concentrations within our cells, and we need to get rid of them. Gluconeogenesis occurs. It is essentially the synthesis of glucose, happens within the liver, and so we pick up these 3-carbon metabolites, 2 of them, at least in this case, and we stitch them together into one 6-carbon glucose. So we're creating glucose. We're synthesizing our own glucose, which is something that typically does not happen in animal systems. So this synthesized glucose can either be utilized, again, it can go back into glycolysis, or be used in this other energy production pathway, this oxygen requiring more efficient energy production pathway, so it can be used immediately, or it can be, if we're probably building up a lot of glucose because there's a lot of these 3-carbon metabolites, so a lot of glucose means that we can't use it up quick enough for our own energy needs, and now our blood glucose level is starting to increase, and that is not a good thing. We have a very specific, very narrow range within our bloodstream of blood glucose concentration, and if it gets too high or too low, that needs to be remedied. If our glucose concentration gets too high, and we'll talk about this as a specific other G word, if you will, we can stitch it together into glycogen. And we'll talk about how that is ultimately done momentarily. If we look at this now, I'll clean this. I'll try to clean this up a bit here. If we look at this process, just the first 2 here, what we see in gluconeogenesis is this direction of the arrow, but we could think of gluconeogenesis as essentially a simple reversal of glycolysis, which takes glucose to our 3-carbon metabolites. They are essentially simple reversals of one another. Not exact reversals, but many of the same enzymes that go into glycolysis catalyze the reverse reactions which stitch those 3-carbon metabolites back into glucose. So let's look at that specific process. If our blood glucose level is recognized as being too high, well, we have to sift out that free glucose and out of our bloodstream and convert it into glycogen. As we said before multiple times, nothing happens or is triggered within biochemistry without some sort of signaling process. And many of these signaling processes, as we saw before, have these 3 commands: start, continue, and stop. Those 3 commands. We saw that in with the messenger RNA production. We saw that with the replication of DNA and so on. And it's no different in the synthesis of glycogen. Something has to turn it on. We have to turn on the process. So our body recognizes that our blood glucose level is getting too high, so something has to initiate that. So we can take that, sift out that free glucose and convert it into glycogen. Note what happens here as we're taking this glucose out of our bloodstream is our blood glucose level now is coming down, and that is what we want. If our blood glucose level is too high, we want to bring it down into the acceptable concentration range. So the processes which trigger these commands of stop, start, continue within biochemical processes are typically carried out by signaling molecules, and they're known collectively as hormones. Hormones can be, we've looked at a few of these back in the lipids chapter, or prostaglandins act as hormones, and we have human sex hormones where the cholesterol-like structures, steroid-like structures, but the second G word that I'm going to be looking at is known as glycogenesis, the genesis, the birth of glycogen. So we can think of that. That is so it's different than gluconeogenesis, which is the genesis of glucose. Glycogenesis is the genesis, of the building, putting together of glycogen, and it is stimulated by a peptide hormone known as insulin. Its signal is to turn on the glycogenesis process, and when the glycogenesis process is turned on, we're sifting out glucose from the bloodstream. So it stimulates glucose uptake into glycogen, which we see right here. So let's turn this process around. Now, when our blood glucose level was too high, we stimulated glycogenesis by using insulin as our peptide hormone, and that started bringing down our blood glucose level. But that won't stop. Glycogenesis will continue until it's told to stop and have another process start. So let's take a look at our third G word now, and they all kind of sound alike, known as glycogenolysis. The last one was glycogenesis. This is glycogen, so we're talking about glycogen right here, over here on the left. Lysis, to cut. You remember that word? Lysis, to break apart, or to snip, to cut, and so on. So we're lysing glycogen, and essentially we're breaking down glycogen back into free glucose. So glycogenolysis is the exact reversal, if you will, of glycogenesis, and that has to happen because we're always getting fluctuations in our blood glucose level. If it gets too high, we have to start glycogenesis and start sifting out that glucose within our blood. But if it gets too low, well, we have to turn that process off, and that we have to stimulate glycogenolysis to break up glycogen into glucose and to start bringing the glucose level back up, and sort of a back and forth sort of thing, so that what we end up with is a very exquisitely maintained blood glucose concentration. These two processes work against each other, but also in tandem, if you think about it, because they allow for edging up or edging down that blood glucose level. Now, just as we had our peptide hormone insulin, which stimulated glycogenesis, we have a couple of different hormones which stimulate glycogenolysis. One could think it is more important. If we need a source of energy immediately in the form of glucose, we have to break down some glycogen. So there's a couple of ways, a couple of different signaling pathways that bring this about. So we have two hormones depending on the situation. If it's sort of a everyday need for free glucose, for everyday energy needs, we have this signaling molecule known as glucagon. Glucagon is used when there's only a moderate need for increased blood sugar. Most of the time, glucagon is our stimulating hormone, and if we need to start breaking down glycogen for a little push of glucose, if you will. Now, if we were in a situation which requires a lot of blood glucose immediately, if we anticipate we're going to need a lot of energy right away, so specifically during sort of a stress or threat situation, well, another peptide hormone, or not a peptide hormone, but a hormone comes into play, epinephrine, also known as adrenaline, that frees up a lot of blood glucose immediately. It's sort of it sort of supercharges the glycogenolysis process, breaking down glycogen very, very quickly to free up a whole lot of glucose very quickly in this sort of this fight or flight mentality. Either way, our body is telling us we're going to need a lot of energy very quickly. So it's basically a supercharged glucagon is adrenaline. So the slide we just saw essentially winds up Unit 12, in which we talked about that second stage of catabolism, that anaerobic process, which breaks one 6-carbon glucose molecule down into 2 3-carbon molecules of pyruvate. This was a slide that we looked at the beginning of Unit 12, reminding us that there are 2 biological pathways towards energy production. We've just finished looking at one of them, and that was glycolysis, but there's 2 processes at work at any given time. So we already went through Unit 12, this anaerobic process occurring in the absence of oxygen, producing relatively little energy. We found that per molecule of glucose, we will produce 2 ATP. So we're going to move forward now into from glycolysis into the next unit of study, Unit 13, describing an aerobic process. Aerobic process means it requires oxygen. We're requiring oxygen. And in comparison to glycolysis, this new energy production pathway produces a lot of energy, very much more energy in the form of ATP from one molecule of glucose than did the energy production pathway glycolysis we saw in Unit 12. We also saw in Unit 12 in glycolysis that sometimes, specifically during strenuous exercise, that this much more efficient process can be limited. It's limited by the rate of oxygen delivery to cells. That's great, and we can always have this very efficient pathway going if we have oxygen in our cells to allow it to go. But the rate of oxygen delivery to cells is the bottleneck in that process. And when we're under conditions of strenuous exercise, this more efficient process essentially cannot keep up. It shuts down, and we were left with that glycolysis pathway, which we found was ultimately unsustainable. So we're going to look a little closer here now at Unit 13. Is called aerobic energy production, meaning essentially that it requires oxygen. We're going to learn a name for it, and we're going to see how that while it produces ATP, that ATP production pathway is different. The ATP production in glycolysis was through what was called substrate level phosphorylation, if you remember that. This is a different type of ATP production. It is a phosphorylation event, ADP being phosphorylated to become ATP, but it occurs through a different chemical process. So let's take a look at that. So let's follow our journey from our 6-carbon glucose. And this is sort of a piece of a slide that we looked at before. This is essentially glycolysis, going from glucose to 2 molecules of pyruvate. And we saw that this catabolism is continually moving carbon to a higher oxidation state. Again, we can do that by counting the number of carbon-oxygen bonds and carbon-hydrogen bonds on the reactant side and the product side of this. And we find that we increase the number of carbon-oxygen bonds and decrease the number of carbon-hydrogen bonds. So that must be an oxidation. We can say generally that carbon in that process is moving to a higher oxidation state. We had made the statement that carbon moving to a higher oxidation state is synonymous with extraction of chemical energy from that fuel molecule. That chemical energy we're going to find are in the form of hydride ions in glycolysis. If you remember in the last unit of study, hydride ions were not a good thing. They were a waste product. And this waste product had to be shuttled away somehow. And it was shuttled away by NAD swooping in, picking up that hydride H- ion and becoming NADH. Remember that because glycolysis required NAD cycling to NADH. It was to pick up that garbage, that H- hydride ion. So it produces, so we produce these hydride ions in glycolysis, but that is a problem that we had to overcome by having NAD swoop in and take care of it. Now, here's the magic of these two energy production pathways. They are not in they are not in opposition to each other. In fact, they work with each other, kind of like a jigsaw puzzle pieces come together, wherein if we look closely now at our glycolysis, we see that our hydride ions are produced and we get two pyruvates, which are considered garbage. This second process now is called the citric acid cycle. Let's see if I can there we go, the citric acid cycle. That actually takes what was garbage before, these pyruvates are garbage, and the garbage suddenly becomes the recycling. We can take the garbage process of glycolysis and use those 3 carbon fragments in this more efficient energy production pathway to catabolize those 6 carbons further into 6 molecules of carbon dioxide. And if we look at this, let's take a close look. First of all, I want to say that while our first energy production pathway was known as glycolysis, our second energy production pathway is known as the citric acid cycle. And we'll talk about that a little bit more in a minute. That's how I'll refer to it. But both processes produce hydride ions, H-. So H- here, which once was the garbage, this new process, the citric acid cycle, takes hydride ions, which are shuttled around as NADH, and uses them to drive ATP formation. So what once was a downfall of glycolysis is now a boon to this next energy production pathway. So now these hydride ions are sequentially removed, but they're used. They're not thrown away. They're not just picked up and thrown away. They're used to power the formation of ATP within this new energy production pathway, which is much more efficient. I would further point out here that just as in glycolysis we could tell this was an oxidative process, let's see, I'll just clean this up a little bit here. We had this oxidative process. How do I know it's an oxidative process and thus extracting of energy? Well, our increase of carbon-oxygen bonds and decrease of carbon-hydrogen bonds. Going from pyruvate to carbon dioxide now, look at this difference. Where we started with 10 carbon-oxygen bonds and 6 carbon-hydrogen bonds, we extract all reducing equivalents out of it, meaning there's no more CH bonds. Look at this. 24 carbon-oxygen bonds, 0 carbon-hydrogen bonds. This carbon dioxide is as oxidized as carbon can get. So from the standpoint of the progressive continual oxidation of carbon and pulling out energy, chemical energy from that, that is as far as we can possibly go. We cannot get further oxidized than, or carbon can never become further oxidized than a carbon dioxide molecule. So because now the production of the hydride ions through both of these energy production pathways are now important, these hydride ions are suddenly important, so because they drive the formation of ATP in this second energy production pathway, we have to have a way of scooping them up, picking them up and being transported to the site of ATP synthesis, which we'll look at the end of this unit of study. So we'll bring our attention back to two of our cofactors in these energy production pathway, enzymatic energy production pathways, that of NAD and FAD. We saw these in the enzymes unit, and we find that NAD is an acronym for nicotine amide adenine dinucleotide. Nicotine amide is this piece right there. That is our vitamin B3, and nature tacks on all this other stuff and it becomes NAD. We saw that back in glycolysis. We needed NAD to shuttle away those, at the time, bad hydride ions, and it would form NADH. Now we find that we can utilize these hydride ions. So NAD is considered one of these shuttles for hydride ions. The next one is flavin adenine dinucleotide, or goes by the acronym FAD. This was our vitamin B2 was this piece right there, and again, nature tacked on a whole bunch of other stuff so that we were able to have these two cofactors in these important energy production pathways. Their sole purpose now is for picking up hydrides. And if we think about what a hydride is, it is H minus. Hydride is 1 proton and 2 electrons, H minus. It is those 2 electrons which are the source of the energy which is going to drive ATP production. So when we talk about hydride ions, the importance of hydride ions, we're talking about the importance of the 2 electrons within that hydride. We call them not just hydrides, but also I'll refer to them as electron carriers. While the hydride is an electron carrier, this NAD and FAD rather are considered electron carriers as well, because they carry the hydride ion. When these two electron carriers, NAD and FAD, pick up a hydride ion, we essentially reduce them. They are both reduced. We saw this with NAD. It is a reduction to NADH. Remember, so here's our 1 CH bond on this piece goes to 2 CH bonds. We've increased in the number of CH bonds, thus it must be a reduction because we picked up that hydride ion. Now, similarly, on our FAD, it also picks up a hydride ion. Essentially, we get more hydrogens in there. If we look at going from, there is no hydrogens here, but we end up with 2 hydrogens over on the right-hand side. So that is considered globally a reduction. And what happens is we have our second electron carrier, FAD. It reduces to FADH2. It is the analog of NADH, NAD to NADH, FAD to FADH2. So we can think of these pieces, or the NADH and FADH2, as laden with a hydride, laden with electrons. They are activated after they are reduced. They have these hydrides which then can be transported to the site of ATP production. Now, this is not a one direction process, but rather, as we saw with NAD to NADH, we can cycle backwards. We can oxidize back to our original electron carriers. So if we follow this along, really NAD and FAD are these giant types of shuttles, if you will, for a simple hydride ion. Through this catabolism of fuel molecules, we produce hydride ions. Well, they're picked up by NAD and FAD to form NADH and FADH2. Well, these two reduced pieces, these NAD and FADH2, NADH and FADH2, then go to the site of ATP production, drop off those hydrides to form ATP, and are cycled back to NAD and FAD, and the process can start over again. So the actual catabolism of glucose, where we're extracting these hydride ions, is not ATP production or much ATP production. It is these hydride ions, also known as reducing equivalents, which we'll talk about, which drive the process of ATP formation at another site, at a different site within the cell, and we'll talk about that a little bit more. So let's summarize. When NAD picks up a hydride and becomes NADH, and when FAD does the same to become FADH2, we call these are laden, if you will, with the hydrides, which carry these very energetic electrons, which will drive the ATP production. So ultimately, in this new type of energy production pathway, this more efficient one, whenever we produce NADH from NAD or FADH2 from FAD, that is energy positive. It isn't ATP energy, but it is like an ATP IOU. Whenever we make one of these things, it will be converted, driven into ATP formation, and then these laden reduced structures will cycle back, will oxidize to back to their original form, NAD and FAD. So we've talked a lot about ATP production inside a cell. And here we come to a difference between our two energy production pathways, that of glycolysis and that of the citric acid cycle. Glycolysis, the energy production pathway glycolysis, occurs freely within the cytoplasm of the cell. It is not specifically carried out inside any cell organelle or anything. It is freely within these reactions occur freely within the cytoplasm of the cell. Now, in contrast, we find that the citric acid cycle and the ATP formation derived from that are relegated to being carried out inside a specific cell organelle. That is a big difference. And these and where the citric acid cycle and its subsequent ATP production are carried out are in cell organelles known as mitochondria. Mitochondria are double-membraned cell organelles, which are considered many times as the powerhouse of the cell. They produce a lot of ATP. Most eukaryotic cells possess mitochondria to produce ATP, not all of them, but most of them, and they are because our cells, or many cells, have mitochondria, and the ATP production which goes on inside of it is much more efficient than the process of glycolysis that we looked at back in Unit 12. When we look at the structure of a mitochondrion, so this is where the energy production pathway known as the citric acid cycle happens, we notice some interesting structural feature. If you look very, very closely here, so there's two membranes in a mitochondrion. There's the outer membrane, but the inner membrane is very, very folded. It's highly folded upon itself if you follow this along. And the more folded it is, allows for a much higher degree of surface area on that inner membrane, and that's going to become important. A higher degree of surface area. Now, what happens in the ATP production inside a mitochondrion happens at this inner membrane. So we have these embedded proteins within this inner membrane which form ATP. If we can maximize the surface area on which this ATP production is being carried out, we can maximize the formation or the rate of ATP formation. So this is why we get that very folded inner membrane within a mitochondrion. Okay, so by the end of this particular lecture, ultimately we're just going to look at the preparation of pyruvate, that 3-carbon pyruvate produced in glycolysis, to be sent into the citric acid cycle. So let's look at an overview of this aerobic respiration, it's called, but what it really is the citric acid cycle. Just to summarize, it uses the 2 pyruvate molecules, those 3-carbon pyruvates produced in glycolysis. Again, remember, in glycolysis, pyruvate was a waste product. It was building up to uncomfortable and toxic concentrations within the cells. We had to initiate fermentations to bring the concentration of pyruvate down so that glycolysis could continue. Well, it turns out now that we have the citric acid cycle to sort of fold into this garbage molecule, pyruvate, becomes the starting material for this next energy production pathway. So it really fits it's like a hand in a glove, one energy production pathway into the other. The garbage is now being used as the feedstock. So this process, as we saw in a general sense, takes these 3-carbon pyruvates and we break them down into carbon dioxide. In this process, which is the citric acid cycle, we produce some ATP directly, but the real gold mine is a lot of production of these reducing equivalents. Remember, I'm calling them reducing equivalents. NADH and FADH2 are now ATP IOUs. Every time you see a production of one of these things, that means great, we're going to be making some ATP. So we can think of them as synonymous with ATP production, whereas back in glycolysis, making NADH was just sort of a pain in the rear end, and we had to deal with fermentation to get it back to NAD so that glycolysis can continue. Now, making NADH and FADH2 is a very good thing. So through this catabolic process in which pyruvate, 3-carbon pyruvate, is converted to 3 carbon dioxide molecules, these electron carriers, NAD and FAD, pick up those hydride ions. They receive those electrons inherent in the hydride ions, forming these reducing equivalents. That's a good thing. So as I said before, these reducing equivalents will cycle back from NADH to NAD and from FADH2 to FAD, but as they do, that will drive ATP formation. So let's look at the first process in preparing 3-carbon pyruvate for the citric acid cycle. A lot of this process in the citric acid cycle, since it's derived from a 3-carbon substrate, that is pyruvate, it's all about watching what happens to the 3 carbons in pyruvate. So over on the left here is our 3-carbon pyruvate molecule, and we'll notice the first thing that happens is we lose a molecule of CO2. We blow off a molecule of CO2, and that is this carboxylate right here. That 1 carbon, and a carboxylate has 1 carbon and 2 oxygens in it. It is carbon dioxide. Indeed, when we exhale, we have a higher concentration of carbon dioxide in that breath. That higher concentration is due overwhelmingly to decarboxylation reactions in energy production pathways. So let's follow this. Our 3-carbon pyruvate blows off carbon dioxide. So we have the first in a 3-step process. That's decarboxylation, loss of a carboxyl group as CO2. And that leads to, or it drives, the formation of an NADH. Look at that. Remember, in glycolysis, the formation of NADH was just a problem we had to deal with through fermentation. Now, the production of NADH is a good thing because NADH is an ATP IOU, and we're going to be eventually caching those NADHs in for ATP production. But so this initial blowing off of one of our carbons of pyruvate as CO2 and driving the formation of NADH leaves us with a 2-carbon fragment. This 2-carbon fragment right here from pyruvate is given over in here, C double bond O, covalently bound to a methyl group, CH3. This is known as an acetyl group. What I've circled here is known as an acetyl group. And that 2-carbon structure is linked up to a much larger structure known as coenzyme A. This coenzyme A covalently bonds to this little tiny acetyl group to give a structure known as acetyl-CoA. And it is acetyl-CoA which will approach the site of the citric acid cycle, and the coenzyme A will dump this 2-carbon acetyl group into the citric acid cycle. So before we move into that actual process, let's look at the actual structure of acetyl-CoA here, because just by abbreviating it as acetyl-CoA, or even showing it like we see right here, that can be deceiving. Let's actually take a closer look at what acetyl-CoA looks like. Getting an actual molecular view of acetyl-CoA brings us back to something I showed you back in the genetics chapter, is that when a molecule needs to be shuttled around, we looked at this already a little bit with NAD and FAD, when a small molecule needs to be shuttled around, typically the shuttle is very, very large. If you remember, transfer RNA was this gigantic L-shaped molecule back in the genetics chapter, and it was carrying around just a tiny little squib on the end of it, which was the amino acid. It was kind of like the analogy of, taking a little jewelry box and you need to drive it to the next town, but you sequester a Mack truck to do that sort of thing. We see that over and over again in nature. Looking at NAD, we get this giant molecule comparatively to take around a little hydride, to pick up a little hydride molecule. So we have this great size disparity in nature, and acetyl-CoA is no different. Let's take a look. This is what we're getting shuttled around, this 2-carbon acetyl group. The shuttle itself is this structure right here. It's gigantic. And again, we see this in nature over and over again in the structure of acetyl-CoA. First thing I'd like to look at how our acetyl group is connected to our coenzyme A right here. That squiggle right there, that means it's a high energy bond, and we saw that before. One feature of high energy bonds is that they are quite labile, very labile. So thus, it's a weak covalent bond, essentially. So it's going to be very easy for coenzyme A to dump off this 2-carbon acetyl group at the site of the citric acid cycle. Very easy, because we have a labile covalent bond. Also, we have a common type of architecture again that we've seen before, and that would be over on the right-hand side here, nucleotide architecture. This is the third time we've seen nucleotide structure in vastly different form. Remember, originally our nucleotide structure was for genetic material, DNA and RNA. A nucleotide which had a nitrogenous base, a structural sugar, and a phosphate. So we saw that as the monomer in the synthesis of DNA, which is in itself a polymer. Now, also we saw nucleotide structure in the structure of ATP. Adenosine triphosphate was also of nucleotide structure. Very different function than information carrying or information storage was. Was essentially a rechargeable battery. That was our energy currency of the cell. Now we see nucleotide structure in a shuttle, a type of shuttle, and that is in the form of coenzyme A. We see these nucleotide structures appearing over and over. Now, again, what the actual task of coenzyme A is it covalently binds to our acetyl group here, and it enters the mitochondria. Now let's think about this. Inside the mitochondria is where this citric acid cycle is happening. But where does the pyruvate come from that fires that citric acid cycle? It is a byproduct of glycolysis, which happens outside the mitochondrion. Remember, glycolysis occurs freely in the cytoplasm of the cell, whereas the citric acid cycle is relegated only to inside these cell organelles, the mitochondria. So this pyruvate itself has to enter the mitochondria, and then it is that 3-carbon pyruvate blows off one of its carbons as carbon dioxide and ends up just as a 2-carbon acetyl group, which is then hooked up to coenzyme A and to become acetyl-CoA, and it's delivered to the citric acid cycle. So let's see if we can summarize that process of going from 3-carbon pyruvate outside the mitochondrion, going inside the mitochondrion and becoming our little bitty acetyl group to be linked up to acetyl-CoA. Okay, in this very generalized summary over on the left, we start outside of the mitochondrion within the cytosol. We have our 3-carbon pyruvate. Remember, that was the garbage produced by glycolysis, which occurs within the cytosol. So pyruvate has to be transported over the mitochondrial outer membrane into the mitochondria, at which point 3-carbon pyruvate loses that carbon dioxide molecule, so we've lost 1/3 of its bulk, really, 1/3 of itself. But in the meantime, what we get from that is we drive the formation of 1 NADH from NADA. That is a good thing. We like that because we're going to take that and we're going to cash it in for ATP down the road. So after that molecule of carbon dioxide is blown off, our large shuttle, coenzyme A, comes in to pick up the remaining 2 carbons on that acetyl group that we see here. And it is connected by way of a thioester bond to coenzyme A to become acetyl-CoA. Moving into the next lecture, we'll look at what the acetyl-CoA actually goes and does. It's going to transport that acetyl group to basically throw it into the citric acid cycle, where we're going to be producing a lot more reducing equivalents and even directly produce some ATP.